Good afternoon. Thank you, Alan, and thank you to ARM for inviting us. I'm here representing MaxCyte. As we heard this morning, let me figure out how to advance this. No? Yes? Perfect. So as we heard this morning in the immuno-oncology panel and the gene editing panel, really one of the key challenges in developing cells as therapeutic products is the ability to control the biology of a cell so that you can pre-program a cell to exhibit a desired pattern of biological activity. So MaxCyte has developed and validated a clinical and commercial scale platform that's rapid, automated, and scalable, that is agonistic as to what the cell is, what the molecules are. We're able to engineer cells for a variety of different applications. And our business model is we really want to be your... I'm going to ask you to help me because this isn't working. The green arrow, okay, perfect. So our business model is we want to be your preferred marketing partner or your preferred delivery platform. So when one thinks about what are means in which you can control the biology of a cell responds to stimuli which comes in from the environment. Could be soluble factors, could be interactions with this other cells, could be gene regulation in the nucleus, could be cell material interactions. However, if you can think about what are opportunities where one can pre-program a cell, all of the methods, the means for available today, target a rate-limiting step inside the cell, which is many steps downstream of where the signal comes in. So if you could actually engineer the cell inside it in a pharmaceutically relevant manner, you would have a more robust control mechanism. And why is this critical? Because if you think of a cell as a complex of 30,000 different genes, only about 25% of those are expressed on the cell surface and lend themselves in the conventional pharmaceutical sense to being druggable targets. Whereas majority of molecules available are actually resident inside the cells. So if there's a means for actually controlling and modulating them inside, that would solve one of the major problems in being able to pre-program a cell. So any platform that allows you to now control the biology by controlling molecular flux inside the cell needs to be non-toxic, specific, reproducible, scalable. It needs to lead to significant enhancement in the biological attributes or skew the therapeutic index. And it needs to be such that it can be deployed in GMP and regulatory compliant manner for ease of manufacturing with rapid ability to manufacture and due considerations for cost of goods. So that's really the design criteria around which the MaxCyte flow electroporation platform was discovered, developed, and has been implemented in a variety of applications to date. Very simplistically, we think of ourselves as cell engineers, not simply as an instrument provider. The reason being, our focus is not just to provide an instrument, but really develop optimized processes which take into account product attributes, manufacturability, consistency, reliability, and scalability. So we can take any cell, autologous, allogeneic, or a cell line, and we can essentially engineer it by loading it with any kind of a molecule, either transiently at the level of the cytosol or permanently without using viruses by using a variety of gene editing tools. So again, we don't care whether there's a long-term permanent change desired or the change is only transient. The biology dictates the process. The platform itself is automated, robust, GMP compliant. It's supported by an FDA master file. It's a fully closed cell processing system. We can run the equivalent of a 200-liter bioreactor in a fully closed manner in about 20 minutes. And we come up with a product that we call an engineered cell that has reliable, reproducible ability where the biology could be pre-programmed and controlled based on a hypothesized mechanism of action. So the technology itself has been deployed in many different applications. So just as a summary and a scorecard, the platform allows us to develop manufacturing processes in compliance with quality, regulatory, GMP requirements. There is no human, animal, recombinant materials anywhere in the cell contacting process. The process is reliable, scalable, is non-toxic, doesn't damage the cells, and leads to consistent patient-to-patient, lot-to-lot ability to control the biology. It's been deployed in many different applications. It's supported by an FDA master file, a master file with the regulatory authorities in Canada, a CE mark, and it's also been used for clinical trials in various other geographies in Asia where master file systems don't exist. We've deployed the technology in over a dozen programs in the clinic with various types of cells for developing novel cell therapy products, and we currently also support one commercially marketed therapy in Japan. I'm going to sort of elaborate a little more in showing you some of the specific examples that we've had experience working with. These encompass a wide variety of cells, including stem cells, various immune cells, dendritic cells, T cells, NK cells, B cells. It also includes gene editing of primary T cells, immune cells, and hematopoietic stem cells. In addition to supporting partnered programs where we deploy our platform as a platform technology in the manufacture of their products, we're also excited to be our own customers for the platform in developing novel proprietary products ourselves. So I'm going to use the remainder of my talk to kind of give you a brief flavor for how we specifically engineer the activity, manufacturability, and come up with, for our partners, new potential biologically effective therapies. And I'm going to use the example of what we call a CARMA product, which is something MaxCyte is developing, which is an mRNA-engineered peripheral blood cell as a means for delivering targeted oncoimmunotherapy for a variety of different indications. Okay. So you've all heard about the excitement with CAR T cells. So very simplistically, why are people excited? So what a CAR T cell is, essentially, it is a T cell or an immune cell into which there's been a GPS inserted that allows the cell to track to where the tumor is and specifically kill the tumor. The reason people are so excited is because the clinical efficacy seen in early indications with B-ALL in CD19 expressing tumors has just been unprecedented. So people have reported now in a couple hundred patients who've been treated, many different groups, 90% plus complete response rates. Never ever seen in oncology with any kind of a drug product ever before, right? So that's really driven the excitement in this field. And that has led to large investments both from investor community as well as large pharma in driving the CD19 CARs into clinical trials. As you heard this morning, however, this process of making CD19 CARs is very complicated. It requires extensive manipulation. It requires control over logistics and supply chain. It requires a 14-day process. It requires a lentiviral vector to manufacture. The challenge beyond just the manufacturing aspects is they are reported to have multiple toxicities, in that these CAR T cells cannot distinguish between what's a normal B cell and a malignant B cell. And so essentially all the patients have B cell aplasia, and they need to be supported by intravenous IVIG infusions to manage the on-target of tumor toxicity of the product itself. In addition, there is the concept of tumor lysis associated cytokine release storm toxicities and some reported neurotoxicities that have also led to potential patient deaths in some of these clinical trials. So the very first question we asked was, can we take learnings from how traditional small molecule drugs and antibodies are developed and apply it to solve the challenges in the CAR T cell field? By making, rather than having a living drug, which is what a CAR-19 is, can we now have an approach where we have a hit-and-run approach where multiple infusions of CAR cells where the CAR disappears over time could potentially lead to durable clinical benefit? And so we asked the question, can you swap out virus by using messenger RNA? And we've now taken this concept, working with academic collaborators, into 8 human clinical trials, looking at different cancers, different target antigens. One of the trials was with mRNA encoding for an antigen called mesothelin, which is expressed in a variety of cancer cells. And some of the published results, these are trials conducted by Carl June's group at the University of Pennsylvania. Some of the results basically have been published, and some were presented at ASCO. Essentially, the summary of it in total is that they're able to effectively manufacture cells. CAR expression is more than 95% in all of the products. We've not seen any on-target of tumor toxicities to date. And these mRNA CAR T cells express CAR over a limited duration, but they are able to home and traffic to areas of tumors. Radiological measurements looking at tumor volumes indicate that in some of the patients we're seeing measurable reduction in tumor volume and tumor burden, which is very exciting. All of these patients have only been dosed with one cycle. They haven't been repeatedly treated to look at durability of the response, right? Because this is a safety study. The other interesting observation which has been reported and published on is in the treated patients, there has been observation of being able to induce the innate and targeted immune responses in terms of what is immunologically called as epitope spreading, which is really the holy grail. So the mechanism is yet to be understood, but it's not just the CAR cell killing the tumor cell, it's killing the tumor cell and then in an environment such that the resident immune system can come pick up shed tumor antigens to generate a broader spectrum immunity against other tumor antigens. So this has now been extended into many other indications. So looking at these, all of these studies, we've never seen any toxicity to date, and we're seeing measurable detection of reduction in tumor burden with limited number of dosing. And so the next question we asked is, okay, so mRNA CAR seems to be an alternative to lentivirus. Can we solve the other challenge in terms of the cost and complexity with cell activation, selection, and expansion? And essentially have come up with a process which we call CARMA, where we take patient cells from a process called leukapheresis, simply isolate mononuclear cells, have CAR be available as an off-the-shelf mRNA molecule, use the MaxCyte platform to load the CAR into PBMCs. There is no selection, activation, expansion, manipulation of cells. It takes us 20 minutes to process a patient's leukapheresis. We wash the cells. A single manufacturing run gives us between 6 and 8 doses that can be aliquoted and cryopreserved for repeat infusions. So essentially, that's the product concept. Okay? Now, why does one need CARMA? I talked about all the CAR-19 programs. So we're not trying to develop a new CAR. We're trying to make CARs be commercially tangible in how they can be manufactured and delivered. So in our thinking, CARMA is really safer because it doesn't restrict you only to CD19. You can go after solid cancers and liquid cancers. It allows for prospective control over on target of tumor toxicity, which is not available, and you have to use suicide switches if you're using lentiviruses. It is easier because the patients are not subject to any lymphodepletion regimens. And it's a simpler regulatory path in terms of how this would be viewed, in terms of the ease of the manufacturing process. Moreover, it's a very rapid process. It takes us a few hours, less than a day, to make a product. And allows for rapid patient treatment, right? So we're not waiting for 4 weeks, 6 weeks, 8 weeks to manufacture the product while the patient's cancer may be progressing and advancing rapidly. So this allows clinicians more timely ability to intervene and provide for effective treatments. And of course, we don't use viruses. The process itself is automated. It's a closed system. It's simple. It's reproducible, can be potentially implemented at point of care in various hospital settings. So significantly reduces the cost and complexity in being able to effectively deliver CAR immunotherapies going forward. So I wanted to use that as an example, but at our hearts, we're really cell engineers. Our goal is to solve two of the biggest problems, irrespective of whether we work with our partners or we're working with our own programs. We want to make cell therapy more effective. We want to be able to pre-program a cell to skew its potency. Okay? The second problem we want to solve is we want to be able to do this in a manner that facilitates ease of manufacturing and reduces the cost of goods from a manufacturing perspective. And at the end of the day, we see of ourselves as really being a platform. And we want to be, going back to the objective, we want to be your preferred delivery platform for all of ex vivo cell therapy applications that you're involved in. So our interest in participating in this conference is to learn more about what you're doing and see if we can help you. We're also actively looking in terms of large pharma and strategic partners, in terms of them being able to help us develop our CARMA product concept for a variety of disease indications as we go forward. So with that, I will thank you for your attention and take any questions outside the room if there are more questions later on. Thank you so much.